Hi class. Okay, I'm going to start this recording from the beginning of this lecture. I know we discussed this in class, but just to kind of re-emphasize some of the information, let's just kind of start from the beginning. You guys can fast forward to where we ended in class, which was DNA right before we started RNA. So the first few sections I'll kind of speed through because we've already discussed this in class. Okay, so again, this is the agenda, right? We're going to talk about nucleic acid. We're going to compare DNA to RNA, compare transcription, translation, and then kind of talk a little bit about what happens during mutation. Okay, this is the learning objective we already discussed in the class. So let's start with DNA. So we kind of talked about how DNA is the universal genetic code, right? So the universal genetic code is going to consist of A, T, C, and G, right? A, T. Oh, it's even harder to write on my mouse. Then, okay, we're going to make this a G. So this is supposed to be a C, right? A, T, C, and G is universal genetic code. It carries the information. So like I mentioned in lecture, think of a sentence like THE can be the nucleotide, and it doesn't mean anything by themselves, but if you put THE together, it forms the word the, and the has a meaning for us. So same thing with DNA. A, T, C, and G by itself doesn't exactly translate to any particular meaning, but you put all those different sequences together and they start forming information for like your genes that will eventually become your proteins, right? And the genetic information can be passed, as we mentioned, from you to your offspring through the form of the chromosomes that are then passed into the gametes. And this is your DNA is all held in the nucleus. One thing I want to point out, just a reminder that this is going to be the case for eukaryote, right? So we are eukaryote. We're going to focus with regards to DNA in humans, even though, as we mentioned in class, this is the genetic code. So bacteria is going to have the same universal genetic code. A mouse is going to have the same universal genetic code. Banana is going to have the same. But one of the things I want to point out is that in eukaryote, the DNA is in the nucleus, right? Whereas in prokaryote, as we mentioned in previous lecture, it doesn't have, they don't have a nucleus, right? And so this DNA oftentimes we refer to as the blueprint because it has all this information. This information is going to be important for when we actually need to go into transcription and translation and make it into protein. So we talked about this, but let's just kind of point it out again. What is a nucleic acid? So it's a bunch of nucleotides that are combined together, right? So your A and your T and your C, G and so on, they're combined together and it becomes this polymer of nucleotides. And that can either be DNA or RNA, depending on if you have a T or a U. So the deoxyribonucleic acid, also known as DNA, is going to be double-stranded. So we mentioned before, so let's pretend this is dad's DNA right here. So this is dad's chromosome 1, the paternal chromosome. It's going to have the same information here as it does on this strand. Same information on both strands. They're double-stranded. Why is it double-stranded? We didn't really touch upon this in class. Why is DNA double-stranded? DNA is double-stranded because DNA is your blueprint. It has all that genetic information. The information is vital. So we want to make sure that information doesn't get degraded. So being double-stranded kind of helps protect all that information, basically, right? It protects the information. It's more stable. So we're forming this because we want to protect it. And RNA is less stable. We'll talk about RNA in a little bit. RNA is less stable. RNA can live, it can stay in your cells for some time, but there are certain RNAs that exist for like seconds and then they get degraded. And there's reason why they're formed. Some of it is known, some of it is not known, but RNA compared to DNA is a lot more unstable, right? So DNA is more stable, therefore we observe it as a double strand because that double strand helps protect it and keep it stable and protect that vital genetic information. We also mentioned it's long, right? Your whole chromosome 1 is going to be, all the information on chromosome 1 is going to be on, the DNA is going to be in that strand. Whereas RNA is just going to be a section that you want to copy of it. So as we mentioned, if you think of like chromosome 1 being, let's say, the Hunger Game 1 book, right? Whereas chromosome 2 is the Hunger Game book 2 and 3. And then there's a new one, if you guys don't know, there's a new one that just got released that talks about Hamish story. I forget what it's called, but that one just got released like I think last week. So let's say that one's book number 5 or whatever. So each one of those is a whole, we'll just do the analogy that book 1 of Hunger Game is chromosome 1. We want to go back and reread chapter 3. So chapter 3 would be an example of RNA. But let's say we want to read chapter 3, but I want to read in Mandarin. So I'm going to make a copy of it, but I'm going to copy it in a different language. So I'm going to take that chapter, I put in Google Translate, and now I got a Mandarin copy. That's going to be our RNA. It's going to be shorter, and it's going to be in pretty much the same language, but slightly different, right? They have the same meaning behind it, but it's going to be slightly different in it. Okay, so DNA, let's go back to this. It's double-stranded for stability. It's long because it's all of our information from chromosome 1. It's going to have a deoxyribose sugar. We'll look at the sugar in a little bit, right? And it's going to have thymine. So we kind of mentioned that in class that it has the T. Ribonucleic acid is going to have ribose sugar, so a different sugar, and it's going to have uracil, and it's going to be single-stranded. It's not double-stranded, it's single-stranded, which is one of the reasons why it's a little bit more unstable. And the direction, as we mentioned, matters. So we'll see that again in a little bit. Okay, I'm going to move my head down here. So let's look at DNA. DNA is a double-stranded one. So as I mentioned, has a deoxyribose sugar. So this is the 5-carbon sugar. It has the phosphate group, right? And we mentioned why it has 1 phosphate group here versus 3, and has to do with how the additional energy in the phosphate is going to be used to actually form those covalent bond between a nucleotide and a nucleotide. And then it has a specific nitrogenous base. So it's going to be adenine, guanine, cysteine, or thymine. Thymine is only in DNA, right? So it's double-stranded. So this strand, we're going to pretend this is the paternal DNA. So again, this is from dad, right? Same genetic information on this strand as on this strand. Same genetic information. This is your DNA structure. So your DNA structure, as we kind of mentioned, this is 1 nucleotide. Oh, that was a really bad circle. Right here, this is another one. Let me see if I can draw this one a little better. Oh, not much better. But you can see this is 1 nucleotide. This is second nucleotide. They're held together. I kind of bound them together. Let's look at this one right here. They're held together by a covalent bond, right, between the phosphate and the 5 prime, right, or 5 prime phosphate and the 3 prime is what I should say. So they're covalently bond. I want you to recognize the covalent bond in that you would have to put a lot of energy if you wanted to break these apart. However, the 2 strands or double helix are held together by hydrogen bond. Hydrogen bond is a weak bond. They're not sharing electron. So what I'm getting at here is that you can easily separate the 2 DNA structure. I think my head's in the way again. You can separate the 2 DNA structure from one another, but you can't just you can't just chop up different parts of the DNA easily, right? Because those are covalent bond, which is why you're going to make a copy of it for RNA, because you can't just be like, oh, I want this chapter, I'm just going to take the original book and just rip it out. Like, you know how many people you made mad if you took up their Hunger Game book and you're just like, I should need chapter 3 and just ripped it out? We don't want to rip that. We actually want to make photocopy of it and translate to whatever language we need. Let's say from British to English, British to US English, which actually is a job. I have a friend and her husband gets paid to translate things from US English to British English. I have no idea what he translates, because I mean, there must be some differences enough for him to get a job. But you're going to translate, not translate, we'll say transcribe. You're going to transcribe it to whatever language that you need, but you are not just going to rip up the DNA, right? So this is held together by covalent, the double strand held together by hydrogen bond. Of your nucleotides, you have complementary bonding for the hydrogen bonding, which means that C is always going to combine with G. T is always going to combine with A. You're not going to see T combining with G, for example. And if you notice, it's because the number of hydrogen bond varies. A C and a G is going to make 3 hydrogen bonds. A T and an A is going to make 2 hydrogen bonds. So 3 versus 2, right? So these two guys aren't complementary to one another because they form different amount of hydrogen bonds. So they're not going to be complementary to one another. So the pyranine versus the purine, I'm going to emphasize that what I want you guys to know is more of the complementary bonding versus memorizing the structure. But just recognize that pyranine has 1 carbon ring, whereas purine has 2 carbon rings. So pyranines are your T and your C, and your purines are your A and your G. Your classmates ask whether or not you're expected to memorize, and so you should know more of the complementaries of them due to the bonding. So a question I might ask you would be more of like either you have a sequence C, T, G, what is the complementary of that? So you should recognize the complementary would be like C, A, G, for example. Or I might tell you something like you did your sequencing and you notice that you have 20% T. How many G's do you have? So think about that for a second. How would you figure it out? If you did your sequencing and you realize that 20%, you have 20% T, how many G's do you have percentage-wise? Let's walk that through. So if you have 20% T, that means you have 20% A, right? Because A and T are complementary. So 20 plus 20 means you have 40%, which means that 60% is going to be GC. So if 60% is GC divided by 2, that means 30% of that genome is G, right? Because G makes 30%, C makes 30%, that's 60, A makes 20%, T makes 20%, that's 40, 60 plus 40 is 100%. Okay, I hope that makes sense. So that's the type of question that like I might ask. Let's see if there's anything else I want to point out on here. Yeah, so they form complementary band. So one of the things that I brought up, and I kind of ended it fast, and I just want to kind of talk about it a little bit more, is I was talking about how the complementary and like why is it reversed? Because you have where are those 2 pens that I use in class? One strand, this is dad's chromosome. Dad's chromosome is going to exist in our cells as a double strand. So boom, we make a duplicate copy of it. Meaning that this and this have the same genetic information, but they're in different orientation. So we're going to assume the tip is the 5' end. So the 5' end is on this side, 5' end is on this side. The information is the same on both strands, but they are going to be complementary of one another because you're basically making a photocopy of this strand and putting it over here. So they have the same information. It's just that this is in this direction and this is in this direction. But the pairs, the information between them, because they have the same information, is going to be complement to one another. So they're just going to, wherever you have a C, you'll have a G in the other end. They're double-stranded, again, because this information is vital between here. The nucleotide information is vital, so you want to make double-strand to make this more stable. But when you actually are going to make when you're actually going to transcribe this into RNA, your genes are coded in a single direction. So, and your genes can be, depending on gene, it can be, we won't go into too much, but just recognize that on any strand, if you're looking at just one strand, your gene can be either in this direction or this direction. It's just that on this strand, we're only going to transcribe genes that are in the same 5' to 3', because 5' is the start and 3' is the end. So you have a gene that is the opposite direction, which you can. If you wanted that genetic information, you would actually get it from the complementary strand. So you would actually get it from this strand. That's probably more information than you guys need to know. What I want you guys to recognize is that direction does matter, because your 5' is your start, your 3' is your end, and we're going to assume for simplicity that most of the genes are going to go from 5' to 3' on, let's say, this strand, right? We're going we're going to assume that's the case. I'm just letting you know that in real biology, you can have genes on this strand coding in both directions. But yeah, let's not worry to that detail at this point. Just recognize direction. Point is direction matters. Okay, so let's move on to our next slide. Oops, sorry, guys. This is where we kind of ended in class. So let's continue our discussion. So we're going to look at RNA next. So RNA, really similar to DNA with a few differences. One, it's a different sugar. So it's a ribose sugar. What is the difference between ribose and deoxyribose? Is deoxy means that you're kind of removing the oxygen. So if you go back and look at DNA, this is a hydrogen right here. Whereas in RNA is a hydroxyl. What I want you to recognize between these two is their DNA and RNA have two different sugar. That's the thing I want to emphasize. Two different types of sugar, either deoxyribose sugar or ribose sugar. They both have phosphate groups and they have almost the same nitrogen as base, except for uracil. So instead of thymine that we see in DNA, we're going to replace that with uracil. And RNA for the most part is single-stranded. There are other cases, we're not going to focus on that. As far as you guys are concerned, they are single-stranded compared to DNA, which is double-stranded. So RNA is single-stranded. And RNA, we're going to use it so that we can transfer that information that we're interested in and then translate it. So let's go back to my dumb analogy of the Hunger Games here. We have the whole book. We love chapter, I don't remember what the chapters are, but let's say we love chapter 3 for some reason. Love chapter 3, we're going to convert it and transcribe it into British English because we don't know how to read it in American English. So we're going to transcribe it to British English, and then we're going to make it into a totally different form. We're going to make it into a play. So we're going to translate the words that we read into an actual whole play production. That's the analogy I'm going to stick with for now, right? So RNA is the transcribed version of that DNA. So it's going to be one small section that we're really interested in that contains the genetic information we're in. So most cases, it's going to be a gene, right? The RNA that we transcribe is going to be a gene. We'll talk a little bit later how it's not always a gene, but in this example, we're going to say we're going to transcribe it and it's a gene, and we're going to take that gene and we're going to make it into a certain protein, or take that chapter and make it into a play production. So what is the function of DNA? We kind of discussed this already. DNA is going to store our genetic information, right? It's going to store all the information. It's going to store all of the letters. So we're going to assume these are like letters of the alphabet, for example. It's going to store all of those letters, and it's going to actually form sentences that we're interested in. So it's an instruction for protein, which, and all of these different processes, which is basically instruction for life, and it's going to contain protein building information. So not everything is going to be into protein, but a lot of what we're going to use the DNA is pretty much to make different proteins that we're interested in. And the nucleotide are the monomers that's going to be added together to form this polymer right here. And this polymer is going to be our nucleic acid. So monomer, polymer. Okay, so DNA contains a lot of information. All of the genetic information together, all chromosome 1, 2, 3, 4, 5, all the way to 22, and the sex chromosome, that's going to be your genome. So when you hear like the Human Genome Project, what does that mean? The Human Genome Project was transcribing or getting all the genetic sequence for all of chromosome 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 to 22, and the sex chromosome. So that's the whole human genome. One thing I want to kind of mention is they're trying to increase the diversity, but the human genome, you can actually get the sequences, and the sequences was derived from kind of a mixture of humans, because humans all have the same genes, but they're different variation, right? So, but one of the things that I want to kind of mention, I kind of want you guys to think about this with regards to how we approach science, is that most of the human genome was originally done with white male DNA. And so why does that matter? Just think about the representation of the type of genes and the variation that we see. All right, and this is the case with historically with a lot of science research in general. Is previously, if you look back to early science, a lot of the scientific research kind of focused on white male, meaning that if there's differences that has to do with gender, if there's differences that has to do with different ethnic, a lot of times that was like kind of, I want to say dismiss or not focus on, right? So a lot of the information that we know kind of has this very, this focus on white male. And so we can miss out on some information because it turns out different genders respond differently to different things. Different ethnicity responds different. And so you see this with, how with the COVID-19, I think like you see this a lot, COVID-19, where like you just had certain demographic that responds differently. We have that the case with, let's say Parkinson's disease. Like there are certain ethnicity that are more prone to being able to get Parkinson's versus other ethnicity. You see this with like, with even something like body odor. There's certain ethnicity that have certain mutations that make it so they have less body odor, like let's say less or more body hair, less body hair, like, and so there's definitely genetic variation. And so I just wanted to point that out, that sometimes scientific research, we have to think about how we design it, because if we don't design it and take it into all of this consideration, with regards to gender, age, ethnicity, even regions, like how people respond in the United States might be very different than how they would respond if we went to Europe itself or if we went to Asia, right? Because of the different environmental factor, that it can influence maybe like the decisions that we make later with regards to pharmaceutical, medicine, research, that it can have an impact. So that's my little TED talk. Thanks for listening. But I just want you guys to think that in the back of your head when you're hearing about different scientific research. So back to this. So genome, like I mentioned, is the sum of all of individual genes. And each gene, like you have, let's say this is chromosome 1. For some reason, I love chromosome 1, maybe because it's the first chromosome, so it's an easy one to say. So chromosome 1, as I mentioned, there's certain regions that have genes, right? So this is a genetic region that has this gene. And in the gene, you'll notice that there's exon and intron. Exon and intron. So what does this mean? Well, exons are the part that's actually going to code for the genetic information that we will need later for protein. The introns, we're actually going to remove those. They're not going to actually contribute later on to the actual protein sequence directly. Now, notice I said directly. So we're going to splice these out, and we're basically going to pull and push together the exon and exon. So in the end, when we actually have our gene, let's see, it was E, I, E, O. And we're going to, in the end, we're going to make protein with just E and E. So we're going to remove our I. Okay, so we have EIE, we're going to remove the I, and we'll end up with EE. Exons is only roughly 1.2% of the human genome, meaning all the information that we have, all of that genetic information, genes is 1.2%. What does that mean? Does that mean that all the other stuff that we have is just like useless? Like why do we have so much other information? Well, so exon is the coding region because it codes for the information we actually will need to make amino acid. Non-coding is the part that does not code for amino acid, but it does have a purpose. It's going to function with regards to regulatory. So we're still learning about the regulatory functions. But what do I mean by regulatory? So let's think more like, what does that mean? That means that the introns can decide like how much of that gene is going to be emphasized, what combination. It's going to have all this other information. It's going to regulate how we actually get that section that we're going to make into protein. So it can have a bunch of information like, oh, which molecule, because you need all these different molecules to be involved to actually do the translation. So it might tell a certain molecule, like a certain transcription factor, to come land here, or it might say, you know, to land here, like how hard it wants it to bound to it, like how much does it want it to actually stick to a gene and keep making protein, or how much it wants it to just kind of stick for tiny seconds, make some protein, and then have the protein leave. Like it, so it regulates how we do gene expression. That's kind of what I want you guys to know, is it regulates how and how much and how we go about regulating information, like regulating that information to amino acids. And so that's going to comprise of 98.8% of the rest of the genome. It doesn't mean it's useless. We used to think that it meant that it's useless. We used to call it junk DNA, but it turns out it's not useless. It actually has vital information. And like I said, we're still trying to figure it out, because there's so much information as far as regulation that is still kind of unknown, just because it's so much. It codes for so much information there, right? 98.8% is a lot of information. So what does all that information do? For the purpose of this class, I just want you guys to recognize that it's a non-coding and it has regulatory function. And that regulatory function can vary from increasing the gene expression and actually decreasing the gene expression, or altering the gene expression. So that's what I mean by regulatory. It can change the way that we do gene expression. And exon is the coding region. That's the part that's actually going to make the amino acid later. So your genetic information is done in what's called triplet code or codon. So we're going to so assume that our English language only recognize 3-letter words. We're just going to make a bunch of sentences with 3-letter words. So codon is that information right there. So this is an example, for example, of a codon, right? The big red dog ate the fat fox. So that could be, for example, ATC right here, CTG right here, GTT right here, right? That's going to translate to a sentence, and it's done in triplet form. So our genes have this nucleotide code that's done in 3, and it's going to be the building block for proteins. So proteins, as we learned in macromolecule, when we did macromolecule, is amino acid, right? So amino acids are the monomer, sorry. The amino acids are the monomer. It makes a polymer, which is going to be the polypeptide. And the polypeptide, after it folds, and sometimes they fold multiple polypeptide will fold together and they form that quaternary structure. That's going to be your proteins. And so this information right here is what leads to this protein right here. So this genetic code, remember, it's codon in 3, right? Every 3 codon is equal to 1 amino acid. One of the things I should mention that I did not write here is that this is going to lead to code. This is I'm trying to write code redundancy. And I'll explain what I mean in a little bit by that, but I wanted to get on here. Oh my gosh, redundant. It's really hard to write with a mouse pad, you guys. Redundancy. Okay, so every 3 codon represents an amino acid. And there are start codon right here. Notice the start, this AUG can either be start or a methionine, so it can be both. And there are certain codons that are stop codon. So that means that your protein has a specific length. And it has a clear start and a clear stop. It has a specific length. That's the whole point of the start and stop. So when you make your protein, your the machinery that does this is going to look for that start signal and look for that stop signal in the context of the codon. It's going to look for AUG, and then it's going to start translating that to protein, and then it will continue to translate until it hits one of these stop codon. When it hits UAA, it's like, oh, that's the end of the protein. I'm not going to go further. So if it hits, wherever it hits the stop codon, it's just going to stop. Okay? So let's take a look at pictures. So we'll use a recipe here. I was using the Hunger Game book because I just started reading the prequel, whatever that you would call it. I guess it'll be a prequel. I'm not sure. So I just been thinking about it, but let's go back to the recipe because that's the example I have here. So DNA, genome information, your whole genome information. This is a giant book. You're only interested in, of that whole recipe book, one recipe. You're going to make a copy of that recipe and you're going to stick that recipe on your recipe card, right? So now that is going to be a copy and it's going to be your RNA at this point. So you transcribed it, stuck it in there. So let's say you hand wrote it out. You hand wrote it, it's transcribed, it's going to stick in your recipe. Now you actually want to make the protein. So you're going to take this transcript and you're going to translate this to, so by translating, let's say I read out the recipe to my kid, and she's going to put the recipe together. So I'm going to say, oh, you need the, flour, sugar, and egg, and you're going to need to grab the measuring cup, and right, you need to grab the baking pan. So this can be the recipes, this can be your baking pan, and then we're going to combine all the ingredients together, put it in the tool, put it in the oven, and you're going to end up with a protein. So the amino acids are your building block to make this protein. Your tRNA, which we're going to visit in a little bit, is one of the tools that you need to actually form this protein. So you're going to need tRNA, which we haven't really seen yet, and you're going to need rRNA, and you're going to need some other enzymes as well. rRNA is ribosome, right? So you kind of remember that you're going to need the ribosome as well. So that can be, these are the tools that you need. So your building block, all of your building block, your tools will eventually make you that protein. Okay, so DNA to protein is basically the gene expression, how we express. So gene expression is how does that DNA instruction get carried out to become protein. And the gene is going to have everything from the structure information to the different regulation, the introns, right? All of that instruction and tools of how we're going to regulate by either making a lot of this gene or making less of this gene or whatever. It's going to be in that intron information. And then the exon is going to be the actual code that we need to make the protein. So the central dogma we kind of visit in class, right, is taking that DNA, that blueprint, transcribing it into RNA, translating it into protein. DNA to RNA is transcription. This is going to happen in the nucleus itself. That RNA is going to leave and it's going to get translated in the cytoplasm. So outside the nucleus, gets translated into the actual protein. So DNA to RNA, RNA to protein. Let's kind of visit each one a little bit. So mRNA is a result of transcription. What do we mean by that? Taking DNA and turning it into RNA is called transcription. The resulting strand is the template that's going to be used later to make the protein. That strand is called messenger RNA, so mRNA. It's going to follow the complementary base rule. What I mean by that, remember that DNA, right, has, let's say, A, T, G, right? The mRNA, when we transcribe it, we're going to slightly change it because we're going to go from American English to British English. So we have to use a slightly different alphabet. Well, I don't know what they use. Maybe that's a bad example, but with mRNA, right, we're going to change the T into a U, but it's going to be complementary. So if we're having an A, T, C, we're going to make it into a T, G, what did I say, A, T, C? I don't remember what I just said, sorry. So we have A, T, C, which means that we're going to make that A into T, that T into A, and a C into G. So we're going to end up with a TAG, right? So it's going to be complementary. And it requires that we take the DNA, the section that we're interested in, we're going to open it up. We're going to unwind it, open it up. So let's wind it because it's a double helix, right? We're going to unwind it, open it up, make that copy of that recipe that we're interested in, and then the DNA will rewind when the RNA is done. So the RNA is going to come in and it's going to, there's all of these nucleotides already exist in the cytoplasm. So it's basically going to grab nucleotides in the cytoplasm and put them onto this RNA strand. Remember, RNA strand is going to be shorter and it's going to be a section of the DNA. So it's not going to be the whole, it's not going to be the whole chromosome. It's just going to be one small section, right? Okay, and it's going to be complementary. So we're making, as I mentioned, ATC on the DNA. The mRNA strand is going to say TAG. And so you have your DNA right here, right? And remember, these two strands are a copy of one another, but reversed. So back to our pen example, they're complementary but reversed. The RNA is going to come in and make a small copy, let's say from here to here. Oh, here, we can make this little pencil. And this pencil strand is gonna just do it on one strand. So it's not gonna make a copy of both strands. Let's say it makes this copy. This is gonna be it's gonna be what we call a sense strand. You guys don't need to know that word, but I want to introduce it just that if I say it, you know what I'm talking about. So the strand that's gonna make a copy of, it's gonna be the sense strand, whereas the other one's going to be an antisense strand. So they're complementary to each other. So it's going to take the sense strand and it's going to make a photocopy of it. I'll turn it this way. And it's going to have a transcribed version. This is your mRNA. So your mRNA here is going to be that GCAU. That's going to be this little pencil right here. So now that we have this mRNA can now move into the cytoplasm. So this right here, this whole transcription, is happening right here in the nucleus, right? And then it's going to be transcribed. And then that transcribe is going to come out into the nucleus. And then in the nucleus, we have what's called translation. So now we're going to take that information. And remember how I showed you before? This guy, let's find it. This right here, which is that genetic information, is going to take your mRNA and it's going to start looking for these codon, and that codon is going to be the information that it needs to make the protein. So it's going to take this and it's going to start looking for AUG, right? Because remember we said AUG is the start codon. It's going to go through here and it's going to look for the start codon so it knows that's where it starts breaking that sequence into sentences. The start codon is basically the beginning of that sentence. We'll say here the, right? For the big, I forget what the sentence was, but for that, the was the first word of that sentence. That's what it's going to look for right here. Okay, so one of the things I mentioned is we have exon and intron. So the mRNA is going to remove, before we translate it, it's going to remove those introns. So it's going to take those introns out and it's going to attach them back together. So here we have this strand. This is the DNA. And let's assume that that all of these red sections are the exon and the light salmon color ones are my introns. So what I'm going to do is I'm going to take all of the light introns and I'm going to cut them out. I don't need them. I'm going to cut them out and then I'm going to basically glue back all the red part. So I'm going to glue back all the red part, and then my mRNA, this is when it will be mature. So there's other process too, where you add the cap, the 5' cap, and I'm saying 5' because direction matters. You add the 5' cap and you add the poly A tail. We won't go into that in full detail. Do recognize there is a cap in detail. So RNA is not mature until we remove the introns and we add this cap and we add this poly A tail, right? So you should, that's why I didn't put RNA in the beginning, was because RNA can be made into ribosomal DNA, it can be non-coding RNA, it can be transfer RNA, which is tRNA, it can be different versions of RNA, but the one that's going to be used for translation is mRNA. And what distinguishes mRNA is the 5' cap, the poly A tail, and no introns. So one of the other things that we can control with regards to regulation is which exon we want to keep. So let's say here you have these 3 exons, right? And we're removing these introns right here. But additionally, part of the regulation is actually to be able to remove maybe this exon as well. So we can cut it here and end up with just this exon, number 1, and exon number 3. And this is just part of the regulation. So this is why one gene does not always code for just one protein, because you can actually have these cases in which one set of instruction can actually code for many things. And it's because part of the regulations is you can decide which exon you want to keep to form different isomers. Isomers I, S, O, M, E. Sorry, I know my handwriting is atrocious. Isomer. Okay. So for example, let's say this is our sentence. This is with all the exon. Well, we can drop, for example, red and end up fat and end up with a version of the mRNA that is the big dog ate the fox. Alternatively, we can take this original with all the exon and we can drop red and we can drop, I think here we just drop red. So the big blank dog ate the fat fox. So these are just different versions of that same information where we just decide to keep and drop, keep certain exon and drop other exon. Okay, so mRNA to protein is called translation. So remember, we have all the different codons now, and we're ready to make it into the actual protein. So we're ready to make the actual cake, right? So we can convert all of these nucleotides to a protein language. The protein language is amino acids. So you guys have seen this chart before, right? When we're talking about the codon. But now these codon code for different amino acids. So each one of these is an amino acid. Remember, amino acids are the monomer that's going to make up the polypeptide. So each one of these, and there's some redundancy in that. For example, here you can have CUU code for leucine. You can also have CUC as well as CUA, CUG. Notice that's typically just the last one that can be replaced as either U, C, A, or G. You'll notice it here too. Not always, right? Because here there's only UC code for histine, whereas AG code for glycine. But typically you have on the third position a little wiggle room. So it's called like a wobble hypothesis, where you have a little wiggle room so that you can actually, if you have if you have like, for example, our example is CU. If you have CU, that last one, if there's a mutation, like let's say the original code was supposed to be CUU, if there's a mutation and you have with CUC, you'll still get leucine. So that's one of the ways that the cells have made it so that you can't end up with too much deleterious protein because of that last one being able to have some wiggle room. So this is a tRNA. tRNA stands for translate RNA. And so this is one of our tools. Remember I said we have many tools in order to do translation. So this is one of our tools. And what this guy does is it will grab onto the amino acid on one end, and the other end, it has what's called anticodon. So remember when I talked about sense and antisense, the anti and the sense has to do with complementary. So here, the codon is what's on the mRNA. That's the code for the protein. And the anticodon is complementary to the codon. So it's going to recognize the complementary sequence on that mRNA to bring the protein over. So it's going to carry a very specific amino acid. And that amino acid that it carries is reflective with this table, this table right here, right? So here we're looking at, which one is it? We're looking at guanine, so UUU or UUC, right? So it's looking for that sequence, UUU. Well, in order to find that sequence, it has to be complementary to that. So it up here is going to be AAA. So the AAA will match with the UUU, and when it matches, it'll make it so that that growing polypeptide, all those amino acids, that growing polypeptide, it'll add that amino acid to the growing polypeptide, and then it leaves, and it gets recycled. Each use means it leaves, it goes and grabs another amino acid, and then it comes back, looks for another UUU, and then adds another beanie amino acid to the growing peptide. So here's a better picture. So all of this is happening in the ribosome. So a ribosome, I said, is rRNA, so it's another type of RNA, and a ribosome will actually be rRNA and a bunch of proteins. So you just need to recognize, hopefully, that in order to get translation, that we need three main players, right? We need the mRNA that has a codon. We need the tRNA that's going to grab the different amino acid, carries it to the ribosome RNA. Then you have the ribosome RNA, which is going to be the site of the actual protein synthesis. So what happens is the ribosome RNA, which is technically two parts, is going to come in and it's going to clamp. It's going to find the mRNA, it's going to clamp on the mRNA, and it's going to start reading through the mRNA. When you find the AUG, which is the start codon, that's when we start the process of translation. So once we find that a tRNA that's carrying methionine, because remember how I mentioned to you that the start codon is also methionine. So when it sees the AUG, it will come in and it'll put a methionine. So a protein always starts with methionine as the amino acid. It puts down that methionine, right? And then it's gonna go to the next guy. So imagine like the ribosome is sitting here, and you kind of have an mRNA sitting here, and it's just kind of get pulled onto the next one. The tRNA comes, lands, puts the amino acid over, right? So it brings in the new amino acid. Then you have this amino acid binding to this guy, because this is the growing polypeptide. So this is the growing polypeptide. So it's going to bind onto over here, and then we're going to move it aside. And then this one, which is now empty, is going to, because this will actually, I would say it's better to put it this way. It's actually going to grow this way. So this right here is going to be moved to that guy, and they all slide over one. So when they all slide over one, then this one's going to be empty and it leaves. Right? So here you go. You have this empty one that's going to leave, this growing polypeptide, and this one with the new amino acid. The new amino acid gets placed down onto it, onto the mRNA, and we're going to move the whole polypeptide over one, and then we're going to go poof and slide over to the next location. And then it's going to happen again and again and again until when? Till hopefully when it hits a stop codon. When it hits the stop codon, it's going to know, okay, I'm going to stop right here, and we're done with our polypeptide. So here's the whole scene. So let's walk through it again. So we have our floating amino acids in our cytoplasm. We have a bunch of tRNA that's floating around cytoplasm as well. tRNA will recognize its corresponding amino acid based on the anticodon down here. The ribosome is right here. The ribosome has these different sites. So this site right here is holding on to the growing polypeptide for now. A new one will come down, and it's going to sit in this location right here, because we didn't go into detail of it, but just recognize that there's three slots, right? There's three locations here. So it's going to sit down this location. This whole ribosome is going to shift over, and it's going to bring down a certain amino acid that's based off the anticodon that recognize and is complementary to the codon on the mRNA. And then the growing polypeptide is going to get moved over and attached to that amino acid. And then this guy will now, the whole ribosome shifts over. So this guy will now be empty because it's no longer has the growing polypeptide. So it's going to leave and get recycled and go back up here and search for another amino acid. Meanwhile, this growing polypeptide will just continue to grow. And then when it hits the stop codon, it's going to stop the translation, and it'll stop, and it'll cut, and it'll leave, and you have a finished polypeptide. So pause the video right here, and see, can you transcribe and translate this sequence? So this is very similar to what I would probably provide you on a question on an exam. It's giving you the sequence right here and asking you to see if you can either transcribe it into RNA or, and remember here, I put mRNA, but we're just going to assume that right here, this region has no introns, okay? We're going to assume this is all exon. Okay, so pause the video, see if you can do this, and then I'm going to restart the video. Okay, so hopefully this is what you got, right? So this process is called transcription or transcribe, right? It's called transcription. Actually, I am going to type that in. So hold on, I'm gonna change it right in the slide. Okay, transcription, right? And so this is just complementary, right? So you should recognize that the parental strand, also known as a template strand. I'll just write that real quick because it's going to be a template for how we're going to actually make that RNA copy. So this is the template strand. It's going to be complementary to that. So the T is going to be complementary with A, right? A with U, because this is mRNA, so now we have a U in place of a T. C with G, G with C, and so on, right? So that's transcription. Now we have our mRNA. We're going to look for that starting codon, which is right here, AUG for methionine, and AUG will translate to methionine. So this is where you have the tRNA is carrying the methionine. The code, what is the code that's on, what is the anticodon that's on the tRNA? Hopefully you recognize that the anticodon should be a U, an A, and a C. So that is what is on the tRNA, right? tRNA is carrying a methionine. It has anticodon of UAG. It's going to flow around. It's going to find the AUG codon, and it's going to start doing translation. And it's going to continue, right? It's going to continue in triplicates. So 3, 3, 3, and then it's going to go, oh, UGA. That is one to stop codon, right? We had those 3 stop codon. And so now it's going to be done with its translation, and it's going to stop. So for this example, this is our whole polypeptide. Polypeptide. Oh my gosh. Sorry, you guys. Yeah, you know what I'm trying to write, right? Polypeptide. Okay. So what if there's an error, right? This is how mutation arises. So there could be an error, and one of the sequences gets changed. There's multiple types of error. Sometimes the error can be just a single base pair. Sometimes the error can be like a deletion or insertion. So, and there's different ways to cause it. It can be caused by physical, so like the actual sun UV rays with a lot of energy harming it. It can be a chemical, like something, for example, if you eat burnt, like barbecue food, that burnt ribs can actually be carcinogenic. So it can cause carcinogen, which is causing cancer. So sometimes it can be a virus, like certain viruses can induce mutation. So these are changes in mutation. And so what happens is that if you have a change in mutation, we're going to have a change in our code, right? And by having a change in our code, it's going to lead to a change in the mRNA, which is the genetic code for the protein. So you can end up with a different protein sequence or a different amino acid sequence, which leads to a different protein sequence. And so because it's a different amino acid, it has different properties. Because remember, when we talked about macromolecule, how the R groups can have multiple different types of atoms that it attaches to, right? And atoms can have different properties. Like we saw the difference between a hydrogen, for example, versus oxygen, right? Hydrogen is not very electronegative. Oxygen is very electronegative. So they have different properties to them. And so those different properties can lead to ways that the protein will change, right? We already saw that the polypeptide can fold in certain ways to make hydrogen bonds. So you can imagine there's going to be a difference with regards to hydrogen bond if you have the R group having an H versus if you have an R group that has an OH, right? Just because of the differences in the electronegative can have lead to different folding. So this can lead to a non-functioning protein because the folding is different. And if the folding is different, then the protein has a different structural component to it, which means it can be non-functional. And one thing I want to point out is that not every single time that you get a mutation, right? Mutations happen. Like your body, sometimes it's just a mistake in the mutation itself because of like something like the UV. Sometimes it's just an error when the DNA is being replicated. And not all mutations can lead to something bad, right? You can have a mutation that's neutral and has no impact. Why? Well, remember when we looked at that big chart of amino acids, right? You can see that for the one that we were looking at, which was, oh, I don't remember. Let's just go back. We were looking at leucine, right? Remember how it has that wiggle room in the end, a U, C, A, or G? So if the mutation, instead of a UAC, it leads to a UAC, you'll still get the same amino acid, right? You'll still get leucine as the amino acid. So it could be a neutral mutation where there's no difference because it still leads to the exact same protein. You can have a point mutation. And so this could be just a switch in the nucleotide, and that might not, that can and cannot make a difference. So in this one where it's a point mutation, this one definitely made a difference because it's glycine, right? But you can have a point mutation that is in the third base, and because of that code redundancy, that might not lead to any mutation, or it'll still be a mutation. I misspoke. It doesn't lead to any impact. There's still a mutation, but it doesn't lead to any impact. The one that can have a big mutations is sometimes the shifting, but not always, because it kind of depends on how many nucleotides are added. So you can imagine if it's in the non-coding area and you have 3 more nucleotides, then the part that codes for the genes, they all shift, but they all shift by 3. So the protein sequence is still going to be the same. So this might not lead to any mutation. However, what if you have like one insertion, like you add one extra nucleotide? Well, that one extra nucleotide can lead to problems because imagine our sentence before, which was, oh, it doesn't let me type. Okay, let's just imagine our sentence before. Now I'm just going to do it with 2 words because it's such a pain to write with this. The big, oh gosh, let's just stick with the big. Okay, let's say we added one extra, or we deleted. Let's say we deleted one extra word. So now we shift everything over. So before our protein was the and big. And so if we lose one nucleotide, now we end up with and then we'll assume that we have another word that I just couldn't type because writing is hard. So now we have this sentence, and this amino acid doesn't make, this protein is not going to make as much sense. It's not going to be the same protein, completely different protein, right? On the same side, we can end up inserting something. So let's say we inserted a extra E, right? So then you end up with, I'm going to write up here. So what if we insert it and we have the, so the first amino acid is the same, but then the next one is EBI. And again, this is now a totally different protein. And so this can also lead to an issue, right? And that's going to lead to the stop codon being shifted to a different location. So you might end up ending it early, or you might end up ending it later. So the frame shift, if anything, that if you alter the read frame, oftentimes that will be the biggest one that has possible consequences of being deleterious. Deleterious just means that it's bad, right? But keep in mind, sometimes mutations are good. Sometimes mutations actually can be beneficial because it adds diversity. So in the previous class, we talked about adding diversity via crossing over, independent assortment. This is another way in which not only humans but other animals have adapted. Is these mutations sometimes can lead to new traits that actually can be beneficial. All right, so I'm going to stop it here. I will put together some worksheets. I haven't done it yet, but I'll put together like a worksheet that kind of will help us reinforce this. And this will end it for Unit 2. So Unit 2 was talking pretty much about in the cells itself, right? So we talked about the different organelles, and we talked about cellular respiration, and we talked about mitosis, meiosis, we talked about genetic inheritance, and we're talking about DNA and RNA and protein, how we do transcription translation. So that is all about like inside a cell. That's Unit 2. In our next unit, which we will start on Thursday, but then we'll be continuing after spring break, will be on the body system. So now we're going to get to learn a little bit about our body. So we're going to do the reproductive system. We're going to do the digestive system. I'm trying to remember what else we're doing. We're doing cardiovascular. And there's one more, which is slipping my mind right now. It's on the syllabus, so we can look on. You guys can look on there. Yeah. So let me know if you guys have any questions. Just send me a message, and I am trying to think of ways to help kind of piece all that Unit 2 together. So if you guys have any suggestions or if you have any requests, please let me know as usual. And I will see you on Thursday, okay? And we'll talk about reproductive, which will be, nice because we just finished meiosis, mitosis, and DNA. So kind of it's nice to now piece it together and see how this is all put together with regards to reproduction. All right, see you guys soon.